actually, I wanted to thank Chris for the introduction for my talk, because he was going to give the definitions, so the theoretical background for the very practical issues or the very practical things that I was going to talk about, using a wide range of oxygen concentrations when measuring mitochondrial function with the O2K. So I think we still can skip this. I will not go to the definitions, but we heard a little bit so that hypoxia, normoxia, hypoxia is kind of at least related to being above or below something. So why should we care about it when we measure in the, oh, I'm going too fast. When we measure in our experiment, when we measure in the experimental chamber. One reason, of course, is to study mitochondrial function at physiologically relevant oxygen levels. Another reason is to investigate oxygen kinetics of mitochondrial function. So you want to see how does a function react to different oxygen concentrations. Also, there are some specific samples like permeabilized muscle fibers where there might be a restriction of oxygen diffusion, and you want to prevent a limitation of respiration in these samples. So you might have to increase the oxygen concentration and also prevent inhibition of photosynthesis by high oxygen levels. So here we have the other way around. Photosynthesis, you will have an increase of oxygen, and at some point you might have an inhibition of photosynthesis. When we talk about oxygen concentrations in the O2K chamber, we typically start at air saturation. So we reach air saturation by opening the chamber and by getting a defined gas phase above our liquid phase. If we do this just opening, so we have air in here, we will reach air saturation, which is dependent on the temperature, the barometric pressure, and the oxygen solubility of the medium. In Innsbruck, for example, we typically reach values of about 175 micromolar. And this is, of course, much higher than what you would have in the tissue or even at the level of mitochondria. And there are two ways. Of course, they can then increase. So you go above air saturation. You can reach this by also having an open chamber, but then using oxygen, which you inject into this gas phase. Or you work with a closed chamber and you have catalase in the medium and you titrate hydrogen peroxide. To go below air saturation, you can again have an open chamber and use a gas like nitrogen or hydrogen to reduce the oxygen concentration, or you can, of course, use a closed chamber and titrate a reducing agent like dithionite. With titrations, so dithionite definitely with sample might be problematic as you have side reactions, and even with the catalase and the hydrogen peroxide with sample, you can introduce air bubbles which will disturb your measurements. So using gases is actually advantageous. The disadvantage about using gases that typically you have to use, or you have to have high pressure gas tanks. Either they're installed or in a central way, or you have to have really this gas tank in the lab. And this is quite inconvenient. So it's, first of all, a safety issue. So you have to handle the high pressure so it's kind of complicated. Or you don't even have it in your lab, and then you have to go somewhere else. So we were thinking about having oxygen and hydrogen in the lab, but not in the form of high pressure tank, and it should be always available. So we started to develop an instrument which produces oxygen and hydrogen by electrolysis of water, the oxia. And we have here the proton exchange membrane, so the produced gases are separated going to the outer chambers. So we have two compartments of 32 milliliter, which have either then oxygen or hydrogen, and this is the production. So we have 7 milliliter per minute of oxygen and 14 milliliter per minute of hydrogen. You might think this is not too much, thinking about these big gas bottles, but I can show you a trace when you inject now this oxygen or hydrogen into the gas phase. So in the upper graph, we injected just 2 milliliter of oxygen, and starting at air saturation, it's only really 3 or 4 minutes that you reach levels of 660 micromolar. And here we injected 8 milliliter of hydrogen, and again, starting from air saturation, within a few minutes, you went down to 16 micromolar of oxygen. So we really have a huge range that we can cover by using these gases, injecting them into the gas phase. For oxygen, it's quite straightforward, because you have oxygen, you just increase the oxygen concentration. Fine, perfect. However, when you use hydrogen to decrease, of course, you don't only decrease the oxygen concentration, but at the same time, you will increase the hydrogen concentration. And hydrogen is supposed to have an effect on mitochondrial function. So we did a test using hydrogen in comparison to nitrogen to do transitions from air saturation down to about 25 micromolar. So we have in bluish, so blue or turquoise, we have the oxygen concentration here. This is the protocol. We use permalyzed HEC-293 cells using pyruvate malate, so N-pathway linked substrates. And we started at levels close or near air saturation. And we measured respiration and hydrogen peroxide production in parallel. Hydrogen peroxide production is something which is supposed to be influenced by the oxygen concentration. And then we did several transitions, always at around air saturation and going down to about 25 micromolar. And in the upper graph, you can see a representative trace of the respiration, and in the lower graph, it's the hydrogen peroxide flow. And we measured mainly in the leak state, because this is the state where you will have the highest hydrogen peroxide production. And when we analyzed these data, so we did 3 experiments. It's really hard to use the pointer without tipping on the thing to go further. You can see that we have, first of all, this is the respiration, it's leak respiration. We have an oxygen dependence, which is seen in each experiment. I separated it because the absolute values were slightly different between the experiments. It's normalized per cell. But what is important, if we compare the nitrogen and the hydrogen, there is no difference in respiration. And also for the hydrogen peroxide production, we also see a slight oxygen dependence. And again, we don't see any difference between nitrogen and hydrogen. So I'm actually a little bit faster now. So as a conclusion, we can say that we can generate the hydrogen and the oxygen by Oxia. It's very fast. It's really just a few minutes, and as you need only a low amount of gas, you can do several experiments after each other. Just keep on producing the gas. It's very safe, and you can produce it wherever and whenever you want to. So this is also a disadvantage when you have these high gas pressure tanks. Of course, you cannot pack them and go somewhere with them. I mean, the Oxia, that's about 1.5 kilogram. You don't have compressed gas. You can just take it and go wherever you want to, put it beside your O2K and produce the gas whenever you need it. So no handling of the high pressure gas tanks. And in our experiments, yes, we saw an oxygen dependence of mitochondrial respiration and also H2O2 production in these HEC-293 cells. So it is important to care about the oxygen concentration that you have in the chamber. And at the same time, we didn't see any dependence on the gas that we were using. So from this experiment, it seems that you can really use the hydrogen as well as the nitrogen to decrease the oxygen concentration. But of course, if you have any doubts, just use the hydrogen, and we're happy to see your results in EBEC 2024 in 2 years, whether we can see a dependence specifically of inducing the hydrogen concentration in the chamber. And with this, I would like to say thank you First of all, of course, to Eric, who initiated this project, also to the team who helped, and also to our partner, VGT Electronic, Mike is here today, who put a lot of effort into making this device safe to really avoid hydrogen combustion. And we just yesterday we got the email that we will probably get the certification, so it's really safe. And also to make sure that the gas is not going somewhere, because gases just have the tendency to escape in each direction. And with this, I'm happy to take questions.
There is plenty of time for this interesting work.
Yes.
I can vouch for how damn difficult it is to get hydrogen cylinders, especially into molecular biology labs. Chemistry departments is one thing, but a molecular biology department, my God, it's difficult. And we've tried generating hydrogen in all kinds of different ways. This looks really beautiful. Can you take it down even lower than that if you were to add a catalytic converter so that you could remove any remaining traces of oxygen in there and get strict anoxia? Is that something you would think about doing? Or
You mean that we really reach anoxia in the chamber with
yes.
using the hydrogen?
So if you, I mean, in any glove box, you would have a catalytic converter that would use hydrogen to react with the oxygen to take oxygen down to zero or very low.
Yeah.
That would be a potential. You're taking it down to kind of low micromolar levels. That's
Yes. So we will not reach really anoxia in this. So not by just having the exchange of
I'm just
gas.
wondering, because you'd find a market in me if you could take it down lower. So it's just a thought.
No.
Maybe we find a collaborator in you.
Yeah.
Well, the 16 micromolar that Sabine showed is partly due to a gaseous capillary through the subreservoir. This is 55 millimeters, and if it is full of medium, aqueous medium, then the diffusion is negligible. But with gaseous phase in the capillary, there is back diffusion of oxygen from the air, and you would also see an increase over a longer period of time. So it was not designed to go down to the critical PO2. We go back to definitions of what is hypoxia, functional hypoxia. It was more a starting point where then respiration goes on, and only then mitochondrial respiration can take oxygen down to virtually zero.
No.
Any more questions? Oh, okay, back there.
I actually would like to comment on that, because we have usually worked with nitrogen gas before, and what I saw in the lab is that when we first added the first few milliliters of nitrogen gas, we managed to get the oxygen concentration as low as you showed, so around 20 micromolar. But if I just then let it be stable for a few minutes and then added an extra shot of a few milliliters of nitrogen gas, maybe it would also work with hydrogen gas, I could lower it to about 5 micromolar, so maybe you could go even lower to a more hypoxic state or anoxic state.
You can definitely go lower, and we also did this, especially you have to take care that you inject it slowly and kind of continuously then. So the 60 micromolar are definitely not the lower level, but it's really not anoxia. You will not go down to zero, but yes, in most experiments, you want to lower it down to 10 micromolar, maybe even 5 micromolar, and then you have typically something in there which is supposed to respire somehow, and then of course you can go down to zero with respiration. But yes, we did the same, so if we didn't reach immediately the level that we wanted to reach, then we just added another shot of gas.
Okay, cool, thanks.
Time? Okay, back.
Very interesting. I was wondering whether this new equipment can be used to study hydrogenosomes in other cells.
Yes, of course. I mean, you have pure hydrogen gas. I mean, it's not, we didn't measure the purity exactly, but in principle you have pure hydrogen gas. So yes, I would say yeah. it would be another option to use it.
So we're out of time for the questions now.
Okay.
And thank you. Are we ready for the first presentation, or we're going on to the third one? Chris?
Yes, so thank you very much. I think I already said thank you to the organisers and Eric for the invitation, and I was really happy to see in response to some of my questions, apart from you being shy, that it seems like everyone in this room has some sort of interest in oxygen and, of course, the term that comes with it, which is oxia. And so the talk I'm going to give today is the ABC of hypoxia, which attempts to clearly define hypoxia. Because since we come from so many different disciplines, and work in many different models, we all seem to attach our own meaning to hypoxia, which I think was clearly evident from what we just saw. So actually, my original presentation was to present to you the results of a very small study that I did in Lausanne from an array of colleagues. And actually what I did was I just asked them this very simple question: what is hypoxia? And to my surprise, which I actually had the right to be surprised given your responses, there was actually a unanimous response in Lausanne: less oxygen than normal. But of course, as any good scientist, I would not get carried away with these perfect results. And so what I did was I increased my sample size by randomly recruiting a fifth subject, who at the time did not reply less oxygen than normal, but of course, what is the norm? And despite me being extremely disappointed with participant 5's inability to clearly define hypoxia, the response did clearly highlight probably the most important consideration of all: defining the norm, the reference state hypoxia. Because without this, it is really futile to even begin to speak about less oxygen than normal and, of course, hypoxia. So with this in mind, what we did was we came up with a very simple ABC of hypoxia, which uses three complementary and connected precepts that are A for ambient, B for biological compartment, and C for control of respiration. And it is this ABC of normoxia that I will now walk you through before closing with the ABC of hypoxia, which is of course concerned with deviations below these reference points. So when we discussed and we began to attempt to define normoxia, we pictured things from the perspective, I think, of Ewald Weibel and his infamous pathway for oxygen, where oxygen gradients from the environment to the mitochondrion exist along the pathway for oxygen from the environment to the mitochondrion. And of course, those in the environment are distinguished from those in biological compartments: organs, tissues, cells, and intracellular microenvironments. So therefore, for our ABC, we began with the environment. And very similar to referring to sea level for expressions of altitude, we defined ambient normoxia as a PO2 that prevails in the contemporary environment at sea level. And of course, this is around 20 kilopascals, or can be the equivalent of 150 millimeters of mercury. And we use the SI definition of standard pressure of 100 kilopascals as the reference, barometric pressure, and water vapor saturated air as the reference. If you're interested more in the hydrosphere, of course, the equivalent would be the dissolved oxygen concentration, or Passer pressure, at air saturation of aqueous environments for you. But irrespective of whether you're interested in the atmosphere or the hydrosphere, beyond ambient normoxia is, of course, B, biological compartmental normoxia, which we define as the PO2 in any compartment of a living organism under ambient normoxia in the absence of environmental stress. And of course, This PO2 is a function of aerobic metabolic activity and oxygen transport from the environment to the various compartments of an organism, of course, some of which are shown here on screen, and not surprisingly, you can see that normoxia fluctuates within a range that is specific for that tissue. And so at ambient normoxia, this intracellular PO2, of course, is a function of oxygen demand and supply, such that biological compartmental normoxia varies from aerobic resting activity right up to maximal aerobic activity, or VO2 max, with values in skeletal muscle seeming to range from about 45 to 5 millimeters of mercury. And this means that our biological compartmental normoxia is, of course, this range 5 to 45, which differs somewhat from what we see in the literature when people talk about physiologically induced tissue hypoxia. But within the framework of the ABC, whether these intracellular oxygen pressures are functionally normoxic depends on whether they lie above or below the control region, this term that was coined by Britton Chance back in 1965. And you can see this oxygen control region highlighted here with a little circle, and we can see that the oxygen trace turns sharply to the horizontal. And it is within this region that we see that oxygen is, of course, oxygen pressure exerts control over respiration. So this control region actually connects B to our final precept C, control of respiration, with a reference to, of course, kinetically saturating oxygen pressures. And therefore, normoxic respiration can be defined as respiration at kinetic oxygen saturation, and hypoxic respiration, of course, below this critical oxygen pressure. So based on these categories that I just defined, A, B, and C, the causes for deviations from normoxia to hyperoxia or below to hypoxia are, of course, distinguished, therefore, by 3 categories: ambient hypoxia, biological compartment hypoxia, and critical function-induced hypoxia. So examples of deviations in A, like we already discussed, of course, may be going to hypobaria, where we have a reduction in the pressure, and of course, we then go below our reference for ambient normoxia. Biological compartment hypoxia may, of course, be induced by these ambient conditions, but not necessarily. And whether or not they are functionally hypoxic, of course, then depends whether the pressure is above or below this critical oxygen pressure. And the same logic goes for B and C. But one thing that I would just like to finish on is, of course, that even if we define normoxia here, everything else does not have to be defined in exactly the same way. And it's this what I just want to touch upon very, very carefully. Because even when we had the little short survey, I think everyone touched upon it, that whether it's in the renal medulla, I believe it was, it's very different from what we use in an incubator. And of course, one very hot topic at the moment in research is, of course, the use of non-physiological oxygen levels in ex vivo research, for example, cell culture. And Sabine talked to us there about how perhaps if we use isolated mitochondria, we can, of course, adapt, or we can use any mitochondrial preparation, we can adapt our oxygen regime. And hopefully this ABC of hypoxia can also be used to inform your study design, not just to define and communicate, but whether or not we have someone exercising in normoxia, breathing fraction of inspired oxygen, 21%. I'm wrong here because this actually could be in hypobaria, in which case it would not be ambient hypoxia, but I just clarify that. Then, of course, we have at the tissue level something that is a little bit more relevant, and we may want to perform our cell cultures in this. And of course, whether or not we are above and below this critical oxygen pressure is paramount for whether we are in functional hypoxia or not. But for example, we want to perform experiments if under conditions of functional hypoxia. Of course, we may wish to then combine this with other models. And if we can define clearly at the level of A, B, and C, we of course then can perhaps better translate and understand the results that we will obtain. And so there we have it. the ABC of hypoxia, defined in relation to the normoxic reference state by 3 complementary precepts: A, ambient, B, biological compartment, and C, critical function. So I hope I have convinced you, whilst it's not an easy task, the ABC of framework for defining hypoxia can be used for communication and also for experimental design, irrespective of your discipline. And I'd be delighted to speak to you a bit more about that and discuss it over the next couple of days. And with that, I would like to thank everyone that was really implicated in this ABC of hypoxia as much as I am. Sabine, Christiana, Louisa, Timmy, Nico, Bengt, who sadly is not here, Eric, our funding sources, and of course you for your attention. Thank you very much.
Super nice talk, Chris. Well done. I have a question about the respirometry experiments, for instance, in your permeabilized fibers. It was mentioned already that where the critical O2 starts to become limiting for respiration is already higher than ambient O2 levels. So you always say, well, we need to be above 300, right? Before that, after. So the critical, in your case, your C, your critical PO2 is actually quite high. Now, of course, with your permeabilized fibers, you have also myoglobin that can add oxygen, but it doesn't necessarily, it's dissolved in the same way as making it like a PO2. So is it not the amount of oxygen that is available for the tissue, rather than the PO2 of the tissue, that you should take into account with the critical?
For the critical?
For the critical part.
No, because it is the pressure that determines your kinetics, and this is where, from these kinetics, is where we define this critical oxygen pressure. So I believe that it should be the PO2 that we take into consideration for these sorts of experiments for this critical function hypoxia. But just to come back, you made a very, very nice point. What I did show was humans exercising and, of course, skeletal muscle. But I think that it goes without saying that the gold standard approach for doing anything in relation to oxygen kinetics is, of course, to use isolated mitochondria. Even though we may actually say that permeabilized fibers, we can debate, it's fine. Maybe we might like to say permeabilized fibers retain their structure, contacts between mitochondria and other organelles. Really, for respirometry, is this true? And actually, I think hopefully we'll have a paper that shows that they are all linked in a certain way, but then we must take into consideration when is it the most appropriate model to use, whether it's the permeabilized fiber, or whether it's isolated mitochondria, or even tissue homogeneity.
But in the permeabilized fiber, you also wash out the myoglobin, right, with your permeabilization steps. So you kind of get rid of that oxygen binding capacity. Then you're looking at a similar thing again, but in real life, in the real body, essentially, you have your hemoglobin, your myoglobin that store a lot of oxygen nearby.
And again, this is why we were a little bit mito-focused in this C, let's say. And what we were trying to advocate is not you should do it this way and we should do it that way, but rather a framework. And actually what you say is very nice because you show one of the ways in which we can use the ABC. It's complementary, that in different models we should use different things, just as you alluded to with permeabilized fibers.
Thanks.
Yes, over here. So I have a question. So we work a lot with intact cells in the Ouroboros system. So what is the error that we make by assuming that we can study mitochondria respirometry in intact cells compared to isolated mitochondria? So how is the oxygen pressure changing?
In one versus the other?
Yes.
So in small cells, I believe that we see very similar oxygen kinetics, meaning in these very small cells, of course, we do not have a diffusion limitation per se. However, in a much larger cell type, such as skeletal muscle fibers, of course, there is this oxygen dependence. So your intact cell, if it's a skeletal muscle fiber, is probably not the best, but if it is a small cell, I believe HEX cells is one example, that these are exactly, they are comparable.
Okay, thank you.
So what about ROS flux? Because that's very sensitive to the oxygen concentration. And my recollection of actually probably some of your early work was that at the kind of mitochondrial normoxia, ROS flux is basically undetectable. And what we're normally measuring it at is practically atmospheric oxygen levels, where we can detect it, but we have no idea what it means because it's not physiological. So, but there's a body of literature where we can compare ROS flux as atmospheric pressure. We don't know what it means, or we can fail to detect it at low oxygen tension, in which case you need better detectors and so on. So have you managed to deal with this in terms of ROS flux?
This is a very good question. So I think that it's quite clear, as you say, between ambient normoxic oxygen pressures or dissolved oxygen concentrations, we do see an oxygen dependence in mitochondrial H2O2 flux, for example, from 200 micromolar down to 50. When we get below, of course, we have a little bit more of difficulty to truly determine, but of course there are different ways in which we might be able to do this, chemical background corrections, so on and so forth, but certainly yes, we should take this into consideration. I think better detectors would be, and better probes that are specific for this, etc. And assays are certainly something that we need to think about. But again, for this to come back, I think the isolated mitochondria are the best for determining mitochondrial H2O2 fluxes, for example, and in the context of hypoxia.
Thank you, Chris.
Thank you.
And now we will hear a talk by Stephen Hand.
Well, again, thanks very much to Eric and his great organizational team for the opportunity to present some of our ongoing work on metabolic downregulation, metabolic arrest, and stress. So moving right into the data. So two goals for the talk today briefly would be to emphasize that for decades it's been known that metabolic depression or preconditioning in animals is positively linked to extended survival under environmental stresses like desiccation, which is one that we'll be looking at today. But many years ago, decades, I put together this slide that is emphasizing for anoxia, tolerance to anoxia on the y-axis for a variety of different organisms. These are aquatic organisms, versus the ability of these different organisms to reduce metabolic rate expressed as ATP turnover under anoxia, percent of the aerobic value. And what you see clearly in this logarithmic relationship here is that as you get to organisms that have a profound ability for metabolic arrest, the tolerance to anoxia goes through the roof. And number one are the brine shrimp embryos, Artemia franciscana embryos that I'll introduce in the next slide. But you can see that as metabolic depression approaches the ametabolic state, the tolerance to anoxia can be in the range of years at room temperature. So this sort of relationship, while shown for these organisms, also applies to mammalian cells and systems as well, that we can talk about if you like. And then secondly, under this metabolic shutdown then, in the case of desiccation, stabilization of targeted biological structures during dehydration is also essential for improving biological outcomes upon rehydration and reactivation. Okay, so this is really the star of the story for most of the story, which is the crustacean, the brine shrimp, Artemia, more specifically the embryos, the gastrula stage embryos. And what we're talking about in this slide then is another type of metabolic depression called diapause, where upon release from the adult, these embryos over a period of a few days reduce metabolic respiration rate to less than 1% of the active value for the active embryo. So this diapause state is a programmed arrest of development and metabolism, such that this depression is occurring under normoxia, under euthermic conditions, and under full hydration. It's an internal downregulation. So the question becomes then, how is such a situation possible under full normoxia? And what we have shown over the years is that one of the major predominant aspects to this is that fuel to the mitochondrion is restricted during diapause. So this is orchestrated at multiple enzymatic sites, but to be clear, during this stage of development, metabolism is fueled exclusively by the disaccharide trehalose. And so we should see that trehalase, hexokinase, pyruvate kinase, and particularly pyruvate dehydrogenase complex through a phosphorylation event are downregulated, which is essentially strangling the mitochondrion and preventing carbon fuel from arriving there. Okay, but we've shown very recently, though, that this inhibition can be heightened by restriction of oxidative phosphorylation through the phosphorylation system. The phosphorylation system, as you know, is the ATP synthase, the ANT, and the phosphate carrier as a grouping. And what we're showing here are P to E flux control ratios, in other words, the maximum oxidative capacity divided by the electron transport capacity. And we're showing this for complex 2 substrates, succinate, as well as a combination of NAD-linked substrates plus succinate. And we're showing this for the active embryo versus the diapause embryo. And you can see in both situations, there is a much greater restriction of OXPHOS by the phosphorylation system during diapause. And particularly important is that if you relate this to the concentration of these lysates that are being used, what you see is that at the most concentrated lysate versus their dilutions, you have the greatest depression of the phosphorylation system, which suggests then that there may be some sort of diffusible inhibitor in these lysates. So we've taken a look at that in more detail very recently by looking at long-chain acyl-CoA esters and their ability to inhibit the phosphorylation system of mitochondria, first in these lysates of the embryo. And we can show, if you remove the BSA, which is used oftentimes in very high quantities in preparing these lysates, and you reduce the ADP concentration down to in the range of 1 millimolar, then you can see a very clear effect of the effect on the PE flux control ratio when you add 30 micromolar oleoyl-CoA, which is in physiological range in terms of oleoyl long-chain CoA concentrations for many tissues. But if you don't downregulate a bit, drop down the ADP concentration, this is very dependent on that because the ADP and the long-chain acyl-CoA esters, their impact on the ANT is competitive, all right? But still, it's appropriate to point out that the in vivo ADP concentration is estimated to be an order of magnitude even lower than that. Okay, so it's rather conservative here. So in the early literature, this impact was noted during hibernation in mammalian ground squirrels back in the 70s. And so this is certainly proof of principle that the buildup of these long-chain acyl-CoAs could promote inhibition of the phosphorylation system seen in our lysates. We took this one step further and isolated the mitochondria from these embryos and show that, again, we can see the same impact, the control value in terms of PE flux control ratio, its depression with, in this case, 3 micromolar oleoyl-CoA, much less than used in the lysates, and that when we can reverse this effect very nicely by the addition of 5 millimolar L-carnitine. As you may know, this is due to the fact that the L-carnitine then can accept the acyl group from these long-chain acyl-CoAs and then by the carnitine shuttle, it can move these into the matrix for elimination by beta oxidation. So with a little bit of malate added as well and giving this about 10 minutes and then redoing the P to E flux control ratio, all of these measured with complex 2 substrate, then we return this completely to the control value. So we're very excited about trying future experiments to actually quantify the long-chain acyl coesters in active versus diapause embryos. Okay, so changing gears just a little bit then. As I said, the second goal is that you still need to evaluate protection of macromolecules during water stress. And a couple of key protective agents have received a lot of attention recently. I won't say much about trehalose at this point. That's a non-reducing disaccharide that's been studied as a stabilizer of biological structures in the dried state. But I'll emphasize late embryogenesis abundant proteins. Now, these proteins are found in 4 animal phyla listed here, which also includes the Artemia embryos. But fascinatingly, when these proteins are in the hydrated state, they are intrinsically disordered. When they dry, they assume their, you might call it native conformation, with a large percentage increase in alpha helix. They're targeted to multiple subcellular compartments, including mitochondria, and they function, again, to stabilize various target proteins, lipid bilayers, etc. And these Artemia embryos, believe me, can go to the very dry state as you have these embedded in salt-encased environments here in the Great Salt Lake. Okay. So what we wanted to do was to take these data that have been appearing in the literature for LEA stabilization of target proteins and even cells, and ask the question, can cellular protection by LEA proteins be extended to the whole animal level? To do this, we looked at Drosophila. That organism, of course, is a desiccation-sensitive animal that does not naturally express LEA proteins. So what we did then was we created several fly lines expressing different LEA proteins, as well as a control fly line that expresses green fluorescent protein as a negative control for these trans genes. And then we looked at survivorship as a function of increasing water loss. And if you cut to the chase here and give these data in Kaplan-Meier survivor curves, what you see is that the control values for the GAL4 GFP fly lines are shown here versus the fly lines expressing LEA proteins here. And clearly, there's a much greater ability to survive extended desiccation in the LEA lines. At 50% survivorship, the water loss is around 50%, where it's extended much further out, almost to 80% in the LEA3 lines. So I think we've then been able to show that, in summary, there is a huge metabolic depression during diapause, and it's accomplished primarily by restricting fuel to the mitochondrion, heightened by the inhibition of the phosphorylation system, and that these LEA proteins can improve tolerance to severe desiccation at multiple levels of biological organization, including now the whole organism. I'd like to thank doctoral students in the lab, undergraduate honors students, and wonderful collaborators and support from the National Science Foundation. Thanks much. Do you have any time for questions? Or?
Yeah, we have 3 minutes. Thank
Okay.
you for the talk, and just a few quick questions.
That was very, very nice talk, thank you. In regard to the CPT1 and Armitis, is it known whether that's sensitive to malonyl-CoA as the CPT1 is in mammals, for instance?
Yeah, in terms of that regulation point, we've not studied that yet. It's a very good question. Correct, yeah, but completely understand the germane aspect to that, but we don't know the answer.
Fascinating talk. I was wondering whether a glycerophosphate can be used as a fuel in artemia, because acyl CoAs are non-inhibitors of glycerophosphate dehydrogenase.
Yeah, of course, the tremendous insect system that uses glycerophosphate is very well known, much lower capabilities in these crustacean embryos. So glycerophosphate dehydrogenase is not as a good of a system in terms of mitochondrial, and even malate alone is not a good substrate for these mitochondria, as you see in certain other invertebrates. So it's more classic in some ways.
Hey, yeah. Steve, great talk. So you mentioned in the before you got into the inhibition of respiration by the oleoil-CoA, the kind of classic inhibition of PDH, maybe PFK,
yes,
by
yes.
phosphorylation. Mm-hmm. So I'm assuming that phosphate source is ATP, probably?
Wait, say again. Yeah. The mechanisms for the trehalase pyruvate kinase impact, hexokinase, we've not looked at those under the diapause state yet. We have under an anoxia-induced metabolic depression, where pH is a major player here. But we certainly know thus far that the PDC downregulation is clearly phosphorylation of site 1. And so that's a phosphorylation event. Whether there are any phosphorylation events that are including the downregulation of those yet to be determined. But those sites were identified by careful analysis of metabolic intermediates through the entire pathway, looking at ratios to identify those sites.
Excellent. And so in those states of metabolic suppression, there's still some ATP production. Do you think that this is just very, very low? Is this fatty acid oxidation, or is it?
No, fatty acid oxidation apparently is essentially non-existent in this state, and so it's when metabolism is downregulated as it is here, there is a matched sort of downregulation of ATP production versus ATP consumption. So during diapause, the ATP levels are fairly well protected, even though both of those sides of metabolism have been parallel dropped in a major way. But ATP is not eliminated during diapause over 2 or 3 months. It's maintained. It drops some, but not nearly to the point of being eliminated at all. Yeah.
Thank you, Stephen. Thank you.
Oh, I'm sorry. I didn't see. I'm so sorry. I didn't see.
There is no time. No time.
Between ADP, ATP, and AMP is going on in the system. Do you know?
Well, what we see in diapause as well as in anoxia is what happens, as in many adenylyl kinase systems, is the ATP will go down some and AMP will go up, and the ADP stays roughly constant. That's something we see with the Artemia system as well. But in diapause, the ATP to ADP ratio does go down modestly, but not completely by any means.
Okay, Steve. Thank you. Our next speaker is Tomas Roach, working here in Innsbruck and running too. Okay.
So thank you very much for this introduction. Yes, my name's Thomas. I'm working at University of Innsbruck, not too far from here. And today I will talk about retrograde signaling during high light stress involves reactive carbonyl or reactive electrophile species. So this is the model organism that I've been using for my research, Chlamydomonas reinhardti, maybe new to some of you. It's basically a single-celled flagellar algae, a large chloroplast which is swimming around. And it can grow like most plants photoautotrophically, in which case it's assimilating carbon dioxide in the presence of light. But it can also, as having a lifestyle living in muddy puddles, can scavenge organic acids, and such can uptake molecules such as acetic acid or acetate and use this to supplement its energy requirements. So this brings me to my scientific questions that I will address in this talk. Does the trophic status of these cells affect its level of light stress? Are reactive electrophile species produced under high light stress? And if so, which species? Is res production related to the production of reactive oxygen species, which are likely to lead to lipid peroxidation and the release of such reactive electrophile species, and do reactive electrophile species contribute to light stress signaling? So here, having a quick look at the effect of the trophic status on the photosynthetic efficiency. This is the FE to FM that you can see here. This is an indicator of the efficiency of photosystem 2. And what we can see comparing the different growth conditions here, so low light, and high light with an additional oxygen stress, is that those photoautotrophic cells, which are in the white bars, have a lower efficiency. This is that they have some photoinhibition and that they are likely to be having some damage to the reaction centers. Now, if we have a look at how the cells can protect themselves, this is known as non-photochemical quenching. It's a way that the cells can divert excess energy to heat in a safe way, rather than leading to, for example, the production of reactive oxygen species. We can see that the photoautotrophic cells here have increased this level of MPQ. They're trying to protect themselves from excess light, indicating that they're most likely under some sort of light stress. Now focusing a bit more inside the cells, looking here at the protein level, here a Western blot looking at the levels of glutathione peroxidase, which is known to protect cells from lipid peroxide breakdown products, such as these reactive electrophiles. And what we can see here is that the photoautotrophic cells have a much higher level of these glutathione peroxidases than those grown under mixotrophic conditions. Here we did an oxyblot here to look at the level of protein carbonylation, and also including a mutant which is deficient in MPQ. It cannot safely release excess energy as heat. And what we can see is that the level of protein carbonylation is again higher in those cells which have been in photoautotrophic cells, which are protected from MPQ, is lower than those which are deficient in MPQ, supporting that MPQ is preventing the formation of reactive oxygen species and also has a role in preventing formation of these protein carbonyls. So overall, we can say that the photoautotrophic cells are suffering more from light stress than the mixotrophic cells. They are more photoinhibited, they have higher MPQ, dissipating more energy to heat, they have high levels of GPX, and suffer protein carbonylation. So protein carbonylation can come from many forms, and I'm just going to focus today on this particular form here, where we know that lipid peroxidation is leading to these reactive electrophile species, which through Michael addition can attach these nucleophilic protein residues. So why is lipid peroxidation particularly relevant to photosynthetic organisms? Well, that's because there is a large source of polyunsaturated fatty acids, such as linolenic acid, which have these electron-rich double bonds, which are prone to be attacked by molecules such as singlet oxygen, which is produced during photosynthesis. This leads to lipid peroxidation, and these lipid peroxides are unstable and can then break down either enzymatically or non-enzymatically to release these reactive electrophile species. Some of these names you might know, malondialdehyde, for example, acrolein, and so forth. So what we did is we measured the concentration of these compounds in the cells transferred from low light to high light, comparing mixotrophic and photoautotrophic cells. And what we could see is clearly that the photoautotrophic cells, in white symbols here, have much higher levels of these electrophile species than those grown under mixotrophic conditions. And during high light treatment, the level also increased. Therefore, it seems that the high light stress is indeed increasing lipid peroxidation and the release of these reactive electrophile species, and that the mixotrophy is protecting from this light stress. So now for the rest of my presentation, I'm going to focus on these two particular electrophile species, that's acrolein and hydroxynonenal, which are two species which are increasing in response to light stress. So what we did is we did an exogenous treatment of the cells, first here with acrolein, and what we could see is that acrolein treatment was inducing the production of this glutathione transferase. This is an enzyme which is protecting the cells from electrophile species. And what we could do is we could actually induce the tolerance of cells to singlet oxygen by giving them some pre-treatments with this acrolein. Therefore, we could increase the tolerance through a minor stress of this acrolein. One of the ways that this might occur is through an induction of glutathione. This is an antioxidant which is protecting the cells from reactive electrophile species. And we could see that this level of acrolein here, which is corresponding with the highest level of tolerance to singlet oxygen, was inducing the highest concentration of glutathione. We could also see that this concentration was not really significantly decreasing the viability of the cells, which other higher concentrations were indeed. So if anyone who's working in the field of stress tolerance is probably well aware that there are different levels of stress. For example, there is the alarm, the resistance, and then the exhaustion phase of stress. As cells encounter their first level of stress, they can resist this, and they can in fact increase their tolerance to this stress. And this is what we call as the eustress response, an increase in acclimation to the stress. However, at higher concentrations, they lose this ability to be able to counteract the stress, and they suffer from de-stress, loss of vigor, and viability. And if we look at our glutathione concentrations, this fits very nicely to this eustress de-stress model, where we can see that the cells with the highest tolerance to single oxygen had accumulated this highest concentration of glutathione. So now focusing a bit more on some other potential signaling strategies which may lead to this eustress response. We did a RNA-seq approach, and for this we had two different treatments, or we had two different controls for the different treatments. For a start, the acrolein treatment was used in volatile form, therefore we used solid cultures, and we used the volatility of acrolein to treat our cells. And for highlight treatments, hydroxynonenal treatments, and for singlet oxygen treatments, we used liquid cultures. So we had two different controls for this. We could see that the level of highlight stress was indeed increasing the level of some of our reactive electrophile species, for example, acrolein, hydroxyhexanal, and hydroxynonenal, and indeed the singlet oxygen treatment through this photosensitizer, rose bengal, was also increasing the level of some of these electrophile species, but for example, not all of them. Now focusing on the transcriptional response of these cells and comparing here, or making a correlation between two different treatments, here focusing on the x-axis here, the influence of the highlight treatment, and correlating the influence of the singlet oxygen treatment, we can see that there is a positive relationship in those upregulated genes induced by highlight as to those upregulated by singlet oxygen. But significantly here we have a good correlation of those which are downregulated, so that the genes which have got downregulated transcription by highlight are also downregulated by singlet oxygen, indicating that some of the highlight stress response is coming through this particular reactive oxygen species, singlet oxygen. We then compared, again, the transcriptional response of 4-hydroxynonenal. So this is potentially one of the products of lipid peroxidation via singlet oxygen, and we could again see a very nice relationship between those which are affected by singlet oxygen to those which are influenced by hydroxynonenal, indicating that potentially singlet oxygen stress or singlet oxygen signaling is passing through a reactive electrophile species such as 4-hydroxynonenal. So in conclusion, we can answer some of these questions. Is the trophic growth status affecting the level of light stress? Well, yes. Mixed trophic cells are protected to a certain level from light stress. Are reactive electrophile species produced under high light stress? And if so, which species? Well, many species are, but we could certainly confirm that acrolein and 4-hydroxynonenal were increasing under light stress. Is the production of reactive electrophile species related to ROS production? Well, we think so, at least to singlet oxygen. And can reactive electrophile species contribute towards light stress signaling? Well, we conclude that there is a significant component of high light stress signaling going through reactive electrophiles via singlet oxygen, at least in the species that we are investigating. Which brings me to my acknowledgements, the team that I'm working with in Innsbruck, and our funders, and of course our collaborants in our most recent projects, which we have with Eric. Thank you for your attention.
Questions? Okay, there.
Very nice talk, thank you. I just have a question regarding your measurement of glutathione. Did you measure the total glutathione or the oxidized form?
In this case, this is the total glutathione, so we use DTT to reduce GSSG. And so the concentrations are total glutathione, but yeah.
Thank you.
Yeah, thank you, Thomas. Actually, I have two questions. The first one is, why do you think algae growing in mixotrophy are better protected for highlight? It seems counter to a little bit the other way around, or what I would have expected. And the second question is, what about the other ROSes? So do you think other ROS species like oxygen superoxide, do you think they have a similar effect? Because if I understood correctly, you focus mainly on oxygen, singlet oxygen.
Yeah, okay, so in response to the first question, why is potentially acetate protecting from highlight stress? Inside photosystem 2, there's a bicarbonate which is involved in the electron transfer from plastoquinone A to plastoquinone B. And the redox potential of this bicarbonate is critical for not only the electron flow, but also charge recombination, which leads to singlet oxygen production. It's been shown in various different systems, not just in Chlamydomonas, but you can substitute this bicarbonate with molecules such as acetate. And this is then affecting the charge recombination events. So it's in essence, it's protecting from singlet oxygen production by affecting the level of charge recombination inside photosystem 2. This is one explanation that we actually published in 2013. It could also be something to do with the energetic balance and the fact that the cells may have a higher level of ATP due to the higher consumption of acetate fueling the Krebs cycle, for example. But this needs to be evaluated a bit more. In response to the second question, so why do we focus specifically on singlet oxygen production? Well, personally, I think it's the major reactive oxygen species which is influencing photosynthesis or causing damage. There are some transcriptional studies on hydrogen peroxide, and we see some overlap to our highlight stressed cells, but we see much more of an overlap with those which have been treated with a photosensitizer. So we're pretty, we're sure that the majority of the signaling is going via the singlet oxygen route rather than, for example, the hydrogen peroxide route.
A quick question about the effects of H&E, 4-H&E. A lot of, this one in particular and other aldehydes can, as you, I think you noted early on, bind proteins, form protein adducts. And I saw you looked at the transcriptional effects of high H&E. Have you looked at the, and you can get antibodies for this, protein H&E, protein adducts, which, as you probably know, are known to modulate protein function. Martin Brand's group has shown that they bind to uncoupling proteins, mitochondrial proteins.
So we're currently doing proteomics to tackle this, and we're investigating which particular proteins are being potentially attacked by not only H&E but more acrolein as well. When we look at the concentrations, there's much higher levels of acrolein than hydroxynonenal in our cells, so we think this is probably a more likely damaging species. This is something we're investigating, but thanks.
Any more questions? There is still time. No? Okay, thank you, Thomas.
Thank you.
And our last talk in this session is by Tomas Morosinotto.
Okay, hello everybody, good morning. Thank you, Eric, for putting together this night gathering. I'm actually learning a lot. Since I'm coming from a different field probably than most of people here in the room, I will just go in very quickly on the motivation of our research. So we work on plants and algae. And so the idea is that we are dealing with major challenges that are the increasing population, that is, of course, asking for more food, energy, and materials. And so we are interested in this, using biology to try to solve some of these issues, in particular one biological reaction that Thomas already told us a few minutes ago, that is photosynthesis, where we use sunlight to fix one molecule that is becoming a problem, like CO2, into biomass that we can use for many uses. And of course, by doing that, organisms also produce oxygen that we use for respiration. So without going into detail, all these reactions is fueled by an electron transport reactions that are driven by light. And a byproduct of this reaction is, as I just said, oxygen. So by measuring oxygen, for us is important because it gives us information about how this reaction is working. So how the structure of these complexes and how this works is actually quite well known now, but what we don't know enough yet is how all this is regulated, because plants and algae are working in an environment and they need to respond to all these dynamics by regulating photosynthesis. So because the environment is changing, of course also because the metabolism is always changing, so they need to modulate the electron transport reactions. So what is the issue if these reactions are not regulated? Actually, Thomas already introduced that, is that photosynthesis is dealing with excited electrons that are used for photochemistry. And when we have a lot of these excited electrons, these are an issue because you cannot store excited electrons, but they start doing other reactions and they produce reactive oxygen species. So there is a cost in if photosynthesis is not unbalanced, is that you start producing damaging molecules. As we are all interested in particular in working with algae that are normally unicellular photosynthetic organisms. And the scope is that we want to use these organisms to produce biomass to complement what we are already doing with plants since thousands of years. Actually, algae are particularly interesting because we can grow them with using waste CO2 and wastewater. They differently from plants, they can grow in very nasty materials and waters. And so actually we can design a process for producing biomass with very high sustainability, so recycling wastes into useful products. Another advantage is that we can grow algae in places where plants cannot grow. So we can grow them in the desert, or like we are doing in our lab, we can grow them in the top of our building. So using area that is not suitable for plants production to produce useful biomass. So just a couple of examples. We have been working in the past few years a lot to try to produce, as I said, useful products. Which kind of products? We try to use strains of algae that are very good in producing lipids to make biofuels, even if this is quite a long-term project because it's quite complex to think about producing biofuels. What is probably more promising on the short term is using algae biomass to make food. So already here we have a couple of collaboration with companies where we try to take a problem that is industry production of CO2 and convert this CO2 in some valuable products like food. So what is the scientific issues that we need to address to reach this objective? Is that we can summarize by saying that we take organisms that live in a natural environment like this, probably not always as nice, but in a natural environment, and we put them in some bioreactors that have different form and shapes, but they look more or less like this. So you can already see that the growing conditions are very different when we move from the nature to this industrial environment. And so what is happening, how we can optimize the productivity here, taking an organism that evolution adapted going here. And so one of the things that we need to address is light distribution, because as you can see here, the culture here is so dark green that is almost black. And this means that we have a first layer of cells, the one that are near the surface is absorbing all the light, and most of the culture instead, since the light absorption capacity of these cells is so high, most of the culture is in the dark or very light limited. And so to address this, we are doing genetic engineering. For instance, one of the modifications that we are introducing is producing strains that accumulate less chlorophyll. So the idea is that with the same amount of biomass, we have less light absorption, so the light is going through the culture better. And so the cells that are more shaded by the other, they can still have enough energy to grow. And we demonstrated actually that by doing that, by putting these low chlorophyll strains into photobioreactors, we can actually increase the biomass productivity by 10 up to 20% in the best cases. So moving to us, what is the interesting for us to measure our oxygen? As we said, oxygen is a proxy for photosynthetic activity. It gives us a good idea how photosynthesis is working. And so we are testing these new photobiology models that actually Tony, that is here, did most of the experiments. So if you have technical questions, you can go to him. And so one of the typical experiments is one classical experiment that are called the PI curves. So basically you increase the light intensity in your sample and you follow how the photosynthetic activity changes. And what you get is a curve like this. So you can have, depending on the increasing of the light intensity, you see an increase in photosynthetic activity. You have, when the light is low, you have a limiting, a linear correlation because the light is limiting, but then you reach a saturation because some other factors that are not light becomes limiting for photosynthesis. So here you can see we tested with different organisms, with different species that have very different cell sizes. And one important feature for us is that a difference from the other systems that we had in the lab, we could go down a lot with the cells concentration. And so we were able to build, to measure different PI curves with a very wide range of cells concentration. Here is the summary. So with the lowest, we had some, of course, the signal to noise ratio was worst, but then we reach an optical range of concentration. And when we increase even more the cell concentration, we found that the photosynthetic activity started to go down. And this is because in this condition, the cell concentration was so high that there was shading between the cells. If we compare the two curves directly, we can see here. So here we have a very good curve with the low cell concentration. With the high cell concentration, you see that the shape is different. It means that here the cells, we have too much cells, and so we have some of the cells that are well lighted and the other that are instead a little bit light limited. And this is important for us because this gives us data to assess and quantify the effect of shading. So we can take this data and go back to model and understand better what is happening in these big photobioreactors. The other thing that we can do is that we can directly compare and quantify the effect of our mutants that have less chlorophyll. Here you see one, and you can see that this organism is doing better than the wild type because it has a better light distribution when we have cells with the high concentration. The very last point is that on the very recent experiments where we have been dealing with light fluctuation, that as I said, regulation of photosynthesis is very important to us to understand. Is one of our main biological questions. And here we expose the cells to several cycles of low and high light. And you can see that with multiple cycles, you see that the situation is changing. So we have, with the multiple cycles, you see that we have a decrease in photosynthetic activities. Here you just take the points here, the trends, and you can see that with each cycles we see a decrease in photosynthetic activity. So now we are dealing with the question of this decrease in activity is due to photosynthetic damage or regulation. That's more or less something that Thomas already addressed. And what we found is that by looking to mutants, that at least part of this response is due to regulation. So this is one mutant without a regulatory mechanism of photosynthesis, and you see that this decrease is less clear. So there is some regulation, at least, that is responsible of this phenomenon. And so we are convinced that this will give us the ability to get new mechanistic insights and quantify this phenomena in vivo. Okay, so I think this is all I wanted to say. My time is over, so I just want to thank all the people in the lab and also the companies that support us for this more applied research. All of you for your attention, of course.
Thank you. Excuse my ignorance in algal biology. Don't you think that if you stir the culture, it will give you a better sense?
Yeah, in principle, yes. Also in practical, but you would need to mix so fast that is unpractical.
Okay, thank you.
So actually, this is one of the questions for the engineers design the systems to optimize the system to invest energy to have a optimal mixing by using less energy and whatever. So, but this is something that we need biological data to give them to allow them to optimize the system. But in principle, yes.
Thanks, Thomas. I just had a question about this last figure you showed with the mutant. Do you see that there is an effect on the decrease in photosynthesis at both light intensities or just the higher light intensity?
It's actually both.
So that's indicating that there is also regulation at this lower light intensity that the cells cannot switch off.
But if you have photoinhibition, still you should have a decrease in if you have a photoinhibition, you should have a decrease in photosynthetic activity also at low light after. So if you damage some of the photosystems, you should have some decrease as well.
But this is a mutant in MPQ, so the MPQ is then active also at this low light intensity than the...
MPQ there is still, since MPQ is there is depending on the xanthophyll cycle, so with the time range that we use, we still have some MPQ activated even in the low light.
Mm-hmm. Thanks.
So my understanding is at least one of the subunits in photosystem 2 has the fastest rate of turnover of practically any protein known every 20 minutes or something. I mean, how much can you see that is part of the regulation, or is it simply broken down and not replaced? Is it not relevant to it? I'm just curious.
It is extremely relevant. It's something that to a certain extent is unavoidable because the photochemical reaction the photosystem 2 is doing is so difficult, and so you need so much energy to take electrons out of water, basically, that some damage is unavoidable. Even in very low light, you still see some damage, so you need to have this protein turnover that is very fast.
Is it simply stripped out at low light intensity, just easier not to have it there?
Sorry, I didn't get that.
Can it just simply be removed at low light intensity? If you're not doing photosynthesis, you simply remove the more labile components of the system.
No, you need to replace it, otherwise you lose activity, and then you in the system that we work, they are completely photoautotrophic, so without photosynthesis they don't survive. So they need to replace it. Then you see that in high light they do it faster, but they're still doing it even in low light. There is actually one thing that we'd like to quantify, because, for instance, when we did transcriptomics, if we didn't enrich the nuclear genes, the gene encoding for this protein is a chloroplastic one, and basically the only thing that was expressed was this protein. So it was really amazing. So they are really investing a lot of energy on that. It would be nice to be able to quantify how much energy they are investing in this repair. I'm expecting that is huge.
I have a very small question regarding your respirometry data. You mentioned at the beginning that CO2 is important, understandably. Do you control for CO2, and how do you control for CO2 in your experiments?
We use bicarbonate in the solution to be sure that we don't have a carbon limitation during the experiments, because CO2 is the final electroreceptors, so if we have conditions when we have carbon limitation, then we are going to affect the oxygen evolution activity. Cool. So this is essential, otherwise we get results that are not good. Okay,
thank you.
Thank you again.
So we would like to thank all the speakers for the fascinating talks, and now we should take the group photo, so please gather around the registration area for that.